Good afternoon. I'm very pleased to be here to talk about our study biodiversity patterns and ecological preferences of the phycobionts associated with the lichen-forming genus Parmelia. The genus Parmelia is a widespread genus in the Northern Hemisphere, distributed worldwide in boreal temperate Europe, North America, and Eastern Asia. This ecologically relevant genus has received considerable attention over recent decades. The genus Parmelia belongs to the Parmelian crown of Parmeliaceae and includes 41 currently accepted species. Most of these species are grouped into two main groups, the Parmelia saxatilis group and Parmelia sulcata group. Recently, the species of Parmelia saxatilis group occurring in the Iberian Peninsula were revised, and the topology of the phylogenetic tree were corroborated for this distribution. The Parmelia sulcata group includes 6 originated species, and the Parmelia saxatilis group includes 12 mainly isidated species. Both isidians already are dispersed packages of the fungus and the algae. Several molecular studies on both groups have been published and have revealed the presence of cryptic species. The cryptic species concept was defined as morphological identical species that can only be recognized by molecular data. For example, this study allows us to segregate Parmelia mayi, Parmelia serrana, Parmelia ernstiae, and the new Parmelia rojoi within the Parmelia saxatilis group. Despite the segregations of several cryptic species from Parmelia saxatilis and Parmelia sulcata complexes, these taxa remain the most widespread species in this genus in cold temperate areas of both hemispheres. Contrasting patterns of broad geographical and ecological distribution in some Parmelia species and geographically restricted distribution in some other species of this genus have been documented. However, the phycobiont diversity associated with this lichen-forming fungal species remains unknown. Trebouxia is one of the most frequent lichen symbionts, and it's estimated to be associated with 80% of lichen-forming fungi in temperate regions and more than 20% of all lichen-forming fungi worldwide. To date, 5 major clades have been confirmed within Trebouxia in a multi-locus phylogeny: clade A, clade C, clade S, clade I, and the new segregated clade D. As mentioned before, the phycobiont diversity associated with the genus Parmelia is poorly known. Libet et al. Analyzed the algal ITS region from 11 mycobiont genera of Parmeliaceae and Lecanoraceae, but they did not include Parmelia. They explored the patterns of symbiotic interaction in these lichens based on ecoregion distribution and mycobiont taxonomy. They conclude that fungal specificity and selectivity for algal partners played a major role in determining lichen partnership. Recently, Osuskawa evaluated the genetic variability of phycobionts within the Parmelia omphalodes groups, which include Parmelia discordans, Parmelia omphalodes, and Parmelia pignatifida, all taxa associated with Trebouxia clades, and showed moderate selectivity in their phycobiont choice. PCA results show that Parmelia omphalodes was characterized by the highest bioclimatic variation. Thus, as the host distribution may be greatly influenced by the requirements of the phycobionts, the aim of this study was to assess the Trebouxia biodiversity in the lichen-forming genus Parmelia to better understand their global spatial distribution and ecological preferences. In this study, 159 thalli were collected from 30 locations, including at least morphologically 9 Parmelia species: Parmelia sulcata, Parmelia barrenoae, and Parmelia encryptata within the Parmelia sulcata group, Parmelia submontana, Parmelia serrana, Parmelia ernstiae, Parmelia saxatilis, Parmelia mayi, and Parmelia omphalodes within the Parmelia saxatilis group. The mycobionts were studied by carrying out phylogenetic analysis of the ITS region. This analysis was key to corroborate the accurate species identification and to segregate cryptic species. Newly obtained sequences were aligned with the ITS published in the Backar's previous DNA barcoding study and with Notoparmelia signifera as the outgroup. The tree topology showed 3 major clades: Parmelia fertilis, Parmelia saxatilis group, and Parmelia sulcata group. The relationships among the clades were unsupported. However, they were congruent with previous described phylogenies. The Parmelia sulcata groups included 4 clades: Parmelia sulcata sensu stricto, Parmelia barrenoae, Parmelia fraudans, and Parmelia encryptata. The Parmelia saxatilis group included 7 clades: Parmelia serrana, Parmelia ernstiae, Parmelia submontana, Parmelia saxatilis, Parmelia mayi, and Parmelia omphalodes. We corroborated the accurate lichen species identification and observed the presence of possible cryptic species within the Parmelia saxatilis group, in Parmelia serrana and in Parmelia saxatilis. To estimate putative mycobiont species boundary in the Parmelia dataset, we performed 3 species delimitation analyses. According to this analysis, we estimated the delimitation of 13 species cluster: Parmelia sulcata, Parmelia encryptata, Parmelia barrenoae, Parmelia serrana A, B, C, Parmelia ernstiae, Parmelia submontana, Parmelia omphalodes, Parmelia saxatilis A, B, which is Parmelia rojoy, C, and mayi. 10 of these lineages could be placed in formally described species, whereas Parmelia serrana B, which includes samples from Sweden and Poland, Parmelia serrana C and Parmelia saxatilis C from Spain, are undescribed lineages in Parmelia and probably cryptic species. The genetic diversity of Trebouxia species was screened by using the ITS region and a chloroplast marker. In the case of the ITS region, newly obtained sequences were aligned with the OTUs published by Libet, also with well-accepted Trebouxia species retrieved from the gene bank, and with Asterochloris mediterranea as the outgroup. 15 microalgae species were detected in the lichen thalli study. According to the clade code mentioned for Trebouxia, all sequences generated in this study belong exclusively to clade S and clade I. Chloroplast marker phylogeny corroborated the Trebouxia assignment at clade level S and I, but showed less resolution at species level. According to the 3 DNA species delimitation analysis, putative species boundaries in the Trebouxia concatenated ITS and the chloroplast dataset were estimated, leading to the delimitation of 15 phycobiont species: Trebouxia I02, Trebouxia flava, Trebouxia finflava, Trebouxia SP1, Trebouxia S08, S02, Trebouxia sp2, Trebouxia suecica, Trebouxia S03, Trebouxia sp3a, 3b, 4a, 4b, Trebouxia australis, and Trebouxia angustilobata. 8 of the lineages could be placed in Trebouxia described species, whereas Trebouxia finflava, Trebouxia sp1, sp2, 3a, 3b, 4a, and 4b were newly detected. Distribution and abundance of Trebouxia species were assessed for each Parmelia species to identify possible patterns in specificity and selectivity at species level. High specificity of each Parmelia group for a given Trebouxia clade was observed. Parmelia sulcata, which include Parmelia sulcata, encryptata, and barrenoae, associated only with clade I. In Parmelia saxatilis group, associated with clade S. This could be related with the vegetative reproductive structures, phenomenon that has been observed in cladonia. However, the degree of specificity is different within each group. Since Parmelia saxatilis group, mycobionts were less specific and associated with 12 Trebouxia species. Meanwhile, those of Parmelia sulcata group interact with only 3 Trebouxia species. Several algal species level lineages showed specificity towards a single Parmelia species, as Trebouxia australis, which associate only with Parmelia mayi, or Trebouxia sp4b, which associate only with Parmelia serrana C. Others were not specific towards a single mycobiont. For example, Trebouxia sp1 accept 5 fungal species level lineages, but prefers Parmelia serrana A. This could be due to the fact that the joint dispersal that occurs in asexual reproduction does not imply the maintenance of the same species of microalgae, but once established, the mycobiont can associate with other phycobiont species. To identify the factor that shape the mycobiont partner distribution, we perform variation partitioning analysis. Both analyses explain unexpectedly high proportion of the variability. Climatic conditions, geography, and the mycobiont explain 99% of the variation. The largest proportion of the variation was explained by the mycobiont, 55% of independent effect. The climatic condition explain 42% of the variability, but explain nothing independently. And the variables associated with geography explain 9% of the variability shared with other variables. Climatic conditions, geography, and the phycobiont explain 98% of variation in the phylogeny of mycobionts. The greatest proportion of the variation was explained by the symbiotic partner, and similar proportion of those in the case of phycobionts were detected for climatic conditions and geography. Network analysis have been applied to investigate the topological properties of the mycobiont-phycobiont interaction. Our networks analysis discriminated 7 ecological clusters. 4 of them included 96 of all taxa, which were cluster 1, cluster 4, cluster 6, and cluster 7. To identify the factor that shape these 7 established ecological clusters, where we perform variation partitioning analysis. We analyze the relative contribution of climate, geography, and substrate to the module associations. the response variable explained 58% of the total variation. The largest proportion of the variation was explained by the climatic condition. Substrate explained was the second most important variation, which independently explained 30% of variability, although 7% was shared with other variables. The third variable, geography, accounted for only a small proportion of the variation. We performed PCA analysis to identify which clusters were represented as a function of the climatic conditions. The seven clusters showed indifference relative to climatic parameters, and they are scattered across the hyperdimensional climatic space. However, a slight difference among the clusters were observed when only the major clades 1, 4, 6, and 7 were analyzed in the PCA. In this case, the cluster 6 seemed to be dissimilar in relation to the type of climate. Indeed, this cluster clearly different in preferring cold and humid environments. The climatic conditions of cluster 1, 4, and 7 was not significant. And these are the authors involved in this study. And thank you very much for your attention.